Sex is similar to hunger and thirst in many ways because it involves a pattern of arousal and satiation. It is under the control of hormones, and it's controlled by specific areas of the brain. But it also differs in really important ways from hunger and thirst. It doesn't fit a pattern of homeostatic tissue need. Humans can survive without engaging in sexual activity, and hence they don't require sex for survival, despite any rumors you may have heard. Although humans are similar to other species in that we require sexual behavior and reproduction for our genes to survive to the next generation. In the 1960s, William Masters and Virginia Johnson conducted groundbreaking research on the human sexual response. They were able to study the sexual behavior of hundreds of couples who volunteered to participate in their lab, and they studied thousands of instances of sexual activity. They identified 4 phases of sexual response from their research: the excitement, plateau, orgasm, and resolution phases. The excitement phase is a period of arousal and preparation for intercourse. The plateau phase is where the increase in sexual arousal levels off. At orgasm, rhythmic muscular contractions occur. The last phase, resolution, follows as arousal decreases and the body returns to its previous state. So here is a diagram of the phases of the sexual response cycle. Although Masters and Johnson did not directly study these, more recent research has shown that the autonomic nervous system, as well as other hormones and neurotransmitters, play important roles in this sexual response cycle. So we'll walk through this and I'll talk about the roles that those systems play. During arousal or the excitement phase and during the plateau phase, the parasympathetic nervous system is active because it's maintaining relative relaxation. Dopamine is important for the arousal, and sufficient serotonin is important during this phase or these 2 phases to maintain an aroused but relaxed state. Prior to orgasm, however, the sympathetic nervous system must be activated, and the reuptake of that serotonin back into the neurons must happen for orgasm to occur. So for those taking SSRIs, selective serotonin reuptake inhibitors, this can be very difficult, since this drug actively prevents that reuptake from happening. And this is why sexual dysfunction is a known side effect of taking SSRIs. Now, following orgasm, the release of hormones such as oxytocin and vasopressin for males and the activation of the parasympathetic nervous system contribute to the resolution phase. Masters and Johnson's work was truly groundbreaking. They studied hundreds of couples and thousands of cases of sexual intercourse. They were truly pioneers in the field of human sexuality. However, their work has been criticized on several grounds. For example, the inclusion of a large number of sexually experienced prostitutes, as well as for the lack of inclusion of cultural and ethnic diversity in their participants. But an even greater problem is the volunteer nature of the participants. There was undoubtedly a selection bias for individuals willing to engage in sexual activity while being watched in a lab. So this selection bias may have had a huge impact on their findings. Now, after the resolution phase, males experience something called the refractory period, during which they are unable to become aroused or have another orgasm for minutes, hours, or even days, depending on the male. Now, this refractory period can be shortened in some cases, and there's a phenomenon called the Coolidge effect that describes a shortening of the refractory period. The Coolidge effect refers to a rapid return to sexual arousal when a new partner is introduced. And legend has it that President Coolidge and his wife were visiting a farm. They were touring it separately, though. And Mrs. Coolidge saw a rooster copulating with a hen and asked how often he made it. And the guide said, well, it was several times a day. And the First Lady said, please mention that to Mr. Coolidge. Well, when President Coolidge was told what she'd said, he asked, always with the same hen? And when told that it was a different one each time, he said, tell that to Mrs. Coolidge. Now, the apparent reason for this phenomenon is the dopamine. In a study involving male rats, dopamine levels in the nucleus accumbens rose significantly following copulation with a female when a second female was introduced. The male rat's potential reward center of the brain was stimulated and increased dopamine was produced. And that increased dopamine led to greater sexual arousal and motivation and shortening of that refractory period. So dopamine is crucial for sexual arousal, motivation, and performance in males. And dopamine activity in the preoptic area of the hypothalamus is involved in sexual motivation for both sexes. The preoptic area of the hypothalamus is one of those structures that's active during sexual behavior. The amygdala is also active as well. So the areas of the brain most often identified as active during sexual behavior for humans are the amygdala and the hypothalamus. Activity in the amygdala has been shown to be important, and the amygdala sends projections to the hypothalamus. The hypothalamus plays a role in secreting sex hormones, in autonomic arousal, and in sex drive. Now, undoubtedly, there are many other areas of the brain that are activated during sexual behavior. For example, in the experience of touch or smells, but these are the structures that are most often identified as being active during sexual behavior. Now we'll turn to the biopsychology of sexual orientation and homosexuality. I'll start with some definitions. Homosexual men usually refer to themselves as gay, and homosexual women refer to themselves as lesbian. About 9% of men and 4% of women report engaging in same-sex activity, and in fact, it's believed that same-sex experiences are fairly common, but the term homosexuality is usually reserved for individuals who show a pattern of behavior that's consistent. Bisexuality describes individuals who are neither exclusively homosexual nor heterosexual, and about 1% of the population claims to be asexual with absolutely no interest in sex. So homosexuality is nothing new, but questions about the nature or the basis of homosexuality remain unanswered. Several hypotheses have been generated to explain it, including social. This was once a common belief, and it was often explained by either the mother's or the father's parenting style or early experiences. More recently, the biological hypothesis seems to be what most people center on, and that is that homosexuality is genetic or somehow related to biological factors, generally determined before birth. There is evidence, in fact, that homosexuality is present early in life, and a majority of homosexuals report that they remember feeling very different very early in life. They showed a high rate of engaging in activities that weren't consistent with their same sex, and they preferred other sex companions. So some have speculated the causes are genetic, and yet there really hasn't been a great deal of support for any particular gene. Some have questioned whether it's epigenetic and whether the influence of the environment has an effect on the expression of genes that produces homosexuality. One of the more recent theories for the basis of homosexuality is overandrogenization, or the older brother effect. And this is the idea that the more older brothers an individual has, a male has, the more likely he is to be exposed to excess androgens in utero, and the mother mounts an immune response to those androgens and starts attacking them. Now, there really isn't any clear idea of the mechanism involved in overandrogenization, but recent studies do seem to support this idea, and it's been found that your odds of being homosexual increase by about 30% for every older brother you have. Another theory that was raised that has its origins in biology is that perhaps homosexuals are deficient in androgens or testosterone. Studies that investigated that have found that there is really no difference in androgen levels between homosexuals and heterosexual males. In fact, one of the early treatments for homosexuality based on this idea actually supplemented homosexual men with testosterone, and it did not affect sexual preference at all. In fact, it actually increased homosexual behavior, probably because testosterone is strongly connected to the sex drive. So more recent research supports the idea that hormonal influences that occur during the critical period of brain development, prenatally, may be related to sexual behavior in adulthood. Studies involving rats have shown that changes in prenatal androgen levels are related to adult homosexuality. For example, female rats who are exposed to androgens and male rats who are castrated or deprived of androgens during that critical period of development show not only brain structural changes, but same-sex sexual behavior. So if we refer back to the slide shown when I was talking about sexual dimorphism of the brain, we notice that for the adult female exposed to testosterone, the sexually dimorphic nucleus appears to be more closely related to the adult male's sexually dimorphic nucleus that developed normally. And indeed, the behavior of that female rat as an adult was similar to an adult male, consistent with that rat's brain sex. So this study, as well as many others, have shown that early hormonal manipulation in animals results in same-sex preference later in life, and also brain structure differences. And this has been found in rats, zebra finches, pigs, hamsters, ferrets, and other animals. So research involving males have also shown this similar effect, where if there's no exposure to testosterone during that critical prenatal period, then the brains of these males will not become masculinized. As adults, their brains will resemble more the brain of a female, and their behavior will be more similar to a female adult in sexual behavior. So together, this evidence suggests that sexual orientation may be determined during the critical period for brain sexual development, which for humans is prenatally. And if the brain of the fetus is deprived of testosterone for males, or if the brain of the fetus is exposed to androgens for females, then as an adult, that animal will display homosexual behaviors and show a brain sex that's more similar to the other sex. All of this together suggests that sexual orientation is determined before birth. One other source of evidence supporting this idea is an investigation of the size of the INAH. I referred to this before, again, when we were talking about sexual dimorphism of the brain. But you'll notice that the size of the third interstitial nucleus of the anterior hypothalamus, that is shown to be different in males and females, and usually it's 2 times larger in heterosexual males than it is in heterosexual females, also shows a difference between homosexual and heterosexual males, such that heterosexual males have a larger INAH than homosexual males. In addition to the INAH, there are other structures in the human brain that have shown differences between homosexual and heterosexual males. For example, the suprachiasmatic nucleus and the anterior commissure both show differences in structure between homosexual and heterosexual males. For homosexual women, CAH females, those females who are exposed to an excess of androgens due to the adrenal glands creating those androgens in excess, are more likely to be lesbian or bisexual as adults. This supports the idea that exposure to androgens early in life is related to adult homosexual behavior for females. 2 other lines of research support the idea of the influence of sex hormones early in development, and they are the index to ring finger ratio and responses of click-evoked otoacoustic emissions. So, for female heterosexuals, the index finger has been shown to be significantly longer than the ring finger, whereas in males, the index finger and the ring finger are approximately the same size. Lesbians, however, have been shown to have an index to ring finger ratio that's similar to males and significantly different from females. Otoacoustic emissions are sounds that are generated within the cochlea in response to clicks, and they have been shown to differ between males and females. These otoacoustic emissions for lesbians and bisexuals have been shown to be significantly different from those of heterosexual females and more similar to those of heterosexual males. So both of these findings are consistent with the idea that the brains of homosexual females have been structurally masculinized prenatally. So now I'll be talking about sexual attraction and romantic love. One of the big aspects of sexual attraction is the role of smells or odors. And when we're talking about attraction and smells, we can talk about 2 different things. One, the odors that we are aware of and we consciously smell. And 2, pheromones, which are odors that we don't consciously recognize, but that can have an impact on our behavior and our attraction to others. So for the ones that we do smell, we have a couple hundred receptors that allow us to determine about 10,000 different smells. Those potential smells can either attract us or repel us from potential mates. One of these particular smells is MHC. MHC, or major histocompatibility complex, is the result of genetically determined antigens that are present on a person's cells, and this contributes to your immunity. MHC is determined by a cluster of genes on chromosome 6 of the human genotype. And ideally, you want to partner and mate with someone whose MHC is very different from yours. Because if you can do that, you will maximize the potential of your offspring to survive. And also, the more different your MHC is with your partner, the more sexually satisfied couples report being, and the more fertile they are. So ideally, we want to find someone whose MHC is very different from ours. And thankfully, studies have shown that we are actually attracted to people whose MHC is very different, because humans find a person's MHC, when it's very different from his or her own, as much more pleasant than the MHC of someone who is very similar to them. So moving on to the odors that we aren't consciously aware of, those would be pheromones. Pheromones are airborne chemicals, and they're released by organisms to have a physiological or behavioral effect on another member of that species. Most pheromones are detected by an organ in the nose called the VNO, the vomeronasal organ, and it's a cluster of receptors, and it's located in the nasal cavity. So the VNO picks up pheromones, and then it sends this information to the hypothalamus and the amygdala, specifically the POA in the hypothalamus. And that's the area in the hypothalamus that is highly related to sexual behavior. Some have argued that it's possible that humans no longer have a VNO, and if we have one, that it's not functioning. In an attempt to confirm that humans still do have a VNO, researchers examined the nasal cavities of fetuses and cadavers, and they found that 60 to 90% of people actually do have a VNO. The next disagreement is whether those genes in the VNO are even functioning. We do have behavioral research, though, that shows that pheromones do have an impact on individuals' behavior, and so that suggests that they are functioning. It's possible that instead of the VNO picking up pheromones, that the olfactory receptors, the ones that influence our perception of smell, may also be picking up pheromones. Nonetheless, it's been shown that pheromones do influence behavior. There have been many studies on this. One of the first studies to demonstrate the effect of pheromones describes something called the Wellesley effect. And the Wellesley effect is the phenomenon of a group of women living in a dorm eventually syncing in terms of their menstrual cycle. And it's thought that the reason why their menstrual cycles sync has to do with the pheromones that they release during their cycle. Other studies have taken pheromones from underarm sweat and put them in perfume and had participants rate their sexual behavior and sexual activity while wearing the perfume, and they found that their sexual behavior and activity while wearing the perfume was significantly higher than when not wearing the perfume. Another study found that men rated the smell of t-shirts worn by women as more attractive when the women were in the middle of their menstrual cycles. And the middle of the menstrual cycle is when the female is more fertile. During that fertile phase, pheromones are released. Another study found that men rated the smell of t-shirts worn by women as more attractive when the women were in the middle of their menstrual cycles, which is the fertile phase. They did not find differences in attractiveness for women who were taking oral contraceptives. Oral contraceptives contain hormones that convince the body that it's pregnant. And when females are pregnant, they don't have any reason to ovulate. So hence they don't put out the fertile pheromones. So taking oral contraceptives stops ovulation and the release of those fertility-related pheromones. Other studies have found that men prefer the sweat of women at mid-cycle, so when they were ovulating. And one famous study, the lap dancer study, studied two groups of lap dancers. One group of lap dancers were taking oral contraceptives, and the other group of lap dancers were not. They had them track their menstrual cycles and how much they made in tips each day. And what they found is that for the women who were on oral contraceptives, there was no difference in the amount of tips they made throughout the month. But for the women who were not taking oral contraceptives, the amount of tips they received peaked during that mid-cycle time when they were in their fertile period. So it's very likely that pheromones, even though we may not consciously register them, have an impact on our attraction and our perceived attraction to other people. Now this sheds a bit of light on attraction to others, but what influences falling in love with another person? Totally different subject. So there was a study by Arthur Arun, and he had participants find a complete stranger, spend an hour and a half revealing the most intimate details about themselves, and then simply stare into each other's eyes for 4 minutes without talking. Now this was such a powerful manipulation that many couples felt deeply attracted after this short experiment. And 2 of the couples even got married. So this is an interesting finding, but it doesn't really shed light on why the participants fell in love. Now we can speculate that part of falling in love is evolutionary. It would seem likely that there would be a preference for people who are drawn into intimate relationships, who experience strong feelings in those relationships, and who therefore want to stay partnered in a sexual relationship, because those individuals would be more likely to sexually reproduce and then stay partnered to make sure that their offspring survived and reproduced themselves. Evolution would also favor a preference for those who are particularly sensitive to the pain of a broken heart or the pain of a relationship ending, because individuals more susceptible to those feelings would be likely to stay connected. to avoid that pain, and that would also increase the chance of offspring survival. Going from the evolutionary to the neurochemical, one could look at falling in love and feeling attraction for another person from a totally neurochemical perspective. Kirschenbaum has studied kissing extensively. She argues that the act of kissing is like taking a drug, a powerful drug like cocaine. It can lead to intense feelings of euphoria and desire. It activates dopaminergic systems in the brain. It leads to cravings. It may even transfer testosterone through the saliva. And that may even serve to increase the attraction and the drive of the female. Kissing may even be a way of assessing whether your MHC and the person you're kissing's MHC is compatible. If we're sensitive to the smell of the MHC, then it makes sense that we would prefer the taste of the MHC to someone who is more compatible. Who knows, maybe MHC has something to do with the proverbial sexual chemistry. Helen Fisher is able to take this phenomenon of kissing and the activation of different systems in the brain and place it into her 3 stages of love theory. According to Helen Fisher, romantic love is a drive, and it's evolved to ensure that the human species stays around. She argues that the experience of true love and attraction and lust and attachment, all those things that go into falling in love and staying in love, can be associated with 3 separate systems in the brain. And those systems guide our behavior to falling madly in love. Fisher's 3 stages of love theory begin with lust. And lust is that initial first stage that's driven purely by sex hormones, testosterone and estrogen. It is the thing that drives you towards that other person. The next is attraction. And attraction includes adrenaline, which generates excitement, increased autonomic nervous system activity. Your heart rate goes up, you start to sweat, you get a dry mouth when you're around that person. Dopamine is also crucial in this stage. Dopamine stimulates desire and behavior that may lead to reward, even craving. It triggers an intense rush of pleasure. She argues it's like taking cocaine. In this stage, a person feels higher energy, less need to sleep, and more focused attention on the relationship and that other person. This may be where kissing fits in. Serotonin is also important during this stage. And it may explain why a person finds him or herself constantly thinking about that new partner. It works a little bit the same way it does in people with OCD. In fact, Marazzi wanted to study the levels of serotonin in couples who described themselves as madly in love for less than 6 months. He found that the serotonin blood levels of these individuals were equivalent to OCD patients. So low serotonin is important in the attraction phase, and it keeps an individual focused on that other. The third and final stage, according to Fisher, is the attachment stage. And the attachment stage is associated with the cuddle hormone and the bonding hormones, oxytocin and vasopressin. Oxytocin is associated with deep feelings of attachment and bonding. It's released by the hypothalamus in response to touching. It's released during sexual behavior. It's important in childbirth and breastfeeding. One study even linked greater oxytocin levels with deeply looking in the eyes of a loved one. So higher oxytocin levels are related to having greater trust in others, generosity, and an increase in the ability to feel empathy. Newer findings on oxytocin don't limit the impact of this hormone with positive emotions and cuddling. It's related to both positive and negative emotional interaction with others. Vasopressin is also important in this stage, and it appears to be important in mating and parenting behavior, and it was discovered from prairie voles who form particularly strong bonds. When vasopressin was suppressed in prairie voles, it caused them to reject their partners and fail to protect them or be devoted to them. So it was understood that vasopressin is a very powerful hormone in paternal bonding. It is released after sexual behavior for males, and is thought to encourage bonding between males and their families. So this picture of the brain, taken from Scientific American Mind, is a pretty good indication of the different systems in the brain that Helen Fisher includes in her theory. You can tell by the levers the systems that are activated and the systems that are deactivated in the experience of love. So you can see dopamine's high, oxytocin's high, vasopressin's high, serotonin's low. Remember, because the brain in love needs to be focused on that other person and on that other relationship. Stress hormones are high, the person is alert, the experience of feeling in love reduces your sensitivity to pain, and there are certain parts in the brain that are implicated in the experience of love as well. So the research that Fisher and colleagues have conducted to investigate the structures and the brain systems that are involved in those three different stages or phases of love has generally supported the idea that these different systems are involved. However, what's also clear from the research is that these systems don't work linearly. And in some cases in which she's investigated couples who have been in love for many, many years, multiple systems are at work at the same time. So yes, the systems associated with attachment are active, but also in those same couples, the systems associated with attraction are also active. So what this means is that in the experience of long-term love, these multiple systems in the brain interact to produce those feelings of love. Now, looking at this picture of brain activation during the experience of maternal love and the experience of romantic love, what's clear by looking at the parts of the brain that are shaded by the different kinds of love, there appear to be specific parts of the brain that are active. However, some parts of the brain are active for both types of love. So our understanding of the different parts of the brain and the specific structures that are involved in experiencing different kinds of love is very limited right now. What is clear is that the development and the experience of romantic love is complex and involves interactions between multiple systems, including evolutionary, psychosocial, and neurochemical. And the experience of true love is not something that we totally understand. But if you're lucky enough to experience it, it can be a very rewarding feeling. And hopefully, someday, we'll have a much clearer understanding of how these influences work together to produce that love.